The next most important piece of the muscle contraction cycle is the neuromuscular junction, because the muscles cannot just do this whole cycle with the sarcomeres and all that on their own. They need a signal. They need to be excited by something, and that something is a neuron, a motor neuron that will communicate with them via the neuromuscular junction. We are going to be releasing the neurotransmitter acetylcholine to generate what's called a muscle action potential. A muscle action potential. And so if we look here, right, I'm going to just kind of walk us through this picture here. And then we'll get to some of this stuff here, but let's walk through this. So here is our presynaptic neuron. This is the neuron, and then down here is the muscle cell. That at the end of this axon terminal, right, we have acetylcholine as the neurotransmitter, right? When we get to that, the end here, that action potential has traveled down, it's going to cause the voltage-gated calcium channels to open, flooding calcium into the end of the neuron. That's going to allow the acetylcholine vesicles to fuse with the end and spill that acetylcholine into that synaptic gap, right, in that synaptic cleft. Acetylcholine then will bind with the acetylcholine receptor, right, the nicotinic receptor there, which will will basically open up sodium channels. Those sodium channels, when they open, will depolarize the muscle cell, which will then cause changes right inside of the cell where the T-tubule, basically that entire depolarization event is going to keep moving down, including along those T-tubules. Remember, the T-tubules run down between the two sides of the sarcoplasmic reticulum, and as that depolarization event moves down, kind of like it moves down with an axon, it's going to trigger the sarcoplasmic reticulum to release calcium. When that calcium gets released, then we go into the sliding filament model, where we have actin, in the troponin on the actin, binding with the calcium, moving the tropomyosin, myosin heads grabbing onto actin, power stroke, moving the thing in. This will continue, right? This depolarization, all that will continue until acetylcholine is broken down by the enzyme acetylcholinesterase, which breaks basically acetylcholine in half, right, into the choline and acetate, which then stops the stimulus, which then stops the depolarization of the muscle cell, which then allows the sarcoplasmic reticulum to pump calcium back into itself, removing the calcium from the sarcomere. So if we kind of look at what all is going down, right, so here is our axon terminal, right, as the action potential of the neuron travels down, right, it's going to get to the end, and then it's going to open up calcium channels. Those open, which then causes our acetylcholine vesicles to fuse and spill their contents. The acetylcholine travels across the synaptic cleft, binds with acetylcholine receptors. That is going to then cause the sodium channels to open, which then triggers a muscle action potential because it'll depolarize. All right, acetylcholinesterase will stop acetylcholine so that we don't send another muscle action potential until we get more acetylcholine until we get another nerve impulse to release more acetylcholine. Right, so this is like the entire thing that's going on, right? So this change in the action potential is going to travel along here, right, down through the T-tubule, right? As it gets down through the T-tubule, it's going to trigger the sarcoplasmic reticulum on either side of it to release calcium into the sarcoplasm. And as it does that, right, calcium then finds the sarcomere. It binds to troponin, which causes tropomyosin to move, which allows the actin, the myosin binding sites on actin to be revealed. Right, we can now use ATP to cycle through myosin, grabbing onto actin and pulling everything in towards the middle, shortening the sarcomere. And then when we get to the end of that, right, calcium gets pumped back into the sarcoplasmic reticulum, which means we don't have it bound to troponin anymore. the tropomyosin slides back over those myosin binding sites on actin, and our muscle goes back to relaxing. And we just reset this, over and over and over again every time a muscle contracts, right? We're going through this cycle. You should be able to talk me through the cycle. This isn't too crazy. This is the basics of how all skeletal muscles work. Now, you may be thinking, okay, well, we stopped the contraction, but how the heck do we reset ourselves? How do we get back to normal? How do we relax, right? So we need a couple things. One, the action potential has to stop, right? Acetylcholine has to be removed. The action potential has to stop. The calcium channels on this sarcoplasmic reticulum have to close, and then we need to pump calcium back into this sarcoplasmic reticulum, which is an active transport process, so it's going to take energy. Since calcium's not there anymore, tropomyosin blocks the myosin heads from binding to actin, right? You should be able to talk about this. You should be able to tell me how we stop muscle contraction, because these are the steps involved in stopping muscle contraction. So now let's talk about the motor unit. In a previous video, we talked about motor units being large or small, but it was just basically like, what is the single neuron, the motor neuron, innervating? How many muscle fibers? If it's a large motor unit, then we have a lot of muscle fibers innervated by that neuron. If it's a small motor unit, then we have very few muscle fibers innervated by that neuron. And remember, the bigger the motor unit, the bigger the stronger the muscle, but we don't get as fine-tuned movements, right? So movements that require more detail, smaller, more controlled movements, right? Very fine motor skills require smaller motor units, right? Because then we have more specific control. The smaller the motor units, the more of them we can have in a given space, and that means we can more delicately control them. Whereas a larger motor unit, this is going to control a lot of cells at the same time, so we can produce a lot of force, but we can't, we don't have as many given the density, right, given the space, we don't have as many neurons density-wise controlling that muscle, so we're getting a lot of strength, but we don't get as much absolute control over individual muscle fibers. So there's trade-offs, right? If we need a lot of strength, then a larger motor unit's great. If we need more fine detail, then a smaller motor unit is best. And so here we get into twitch contraction. Right, this is the very brief contraction in a motor unit when we respond to a single action potential. So we're talking about a single action potential. What contraction do we see? Number one, there's a latent period. The action potential and the contraction do not happen exactly at the same time. We need time to respond. So the action potential occurs, and then we have a slight pause before the contraction of the muscle actually happens. So that pause is called the latent period. Then the contraction period is when we have calcium being released, binding to troponin, where we're going through our power stroke, we're going through our sliding filament model, right? And then once we're done with that, right, calcium's back in the sarcoplasmic reticulum, we're relaxing. And then the refractory period is when we have lost excitability. We're not able to be triggered again, right? So this is what the twitch is. It's like this really short, really fast process where a single action potential is generating this pattern. And we can look at what's called wave summation when we have a second action potential. But this second action potential is going to trigger a contraction before the first one's even done. And the second one is going to be stronger than the first. So we call this wave summation. The contractions get stronger by stimuli that are coming in at different times. If we can sustain contraction, even if it's wavering, then we have what's called tetanus. If it's unfused or incomplete, it's wavering. If it's fused or complete, then we can't see individual twitches anymore. It all kind of blurs together. So let's go ahead and talk about this. So here's our single twitch, and there's the amount of force generated with our single action potential. Here's our wave summation, right? We have our first one. We reach the same peak. And before we get back down here, back to start, a second AP comes in, sending the force even higher. This is wave summation. Unfused tetanus. We're getting multiple action potentials before we go back down to normal, leading us to kind of jump, right? We're kind of like bunny hopping up, right? So here's the reaction to the first one, then to the second one, we bunny hop higher, bunny hop to the third, bunny hop to the fourth, right? And so the force is getting stronger with every single action potential, but we can clearly see each individual action potential kind of working on that climb. Fused tetanus, we see that there's a bunch of action potentials. Notice they're getting closer together, and we climb. You can't tell where one action potential started and where one ends, right? Instead of seeing the bunny hop, it's a smooth climb, right? It's slick going up. And so that's what we're talking about here, right? So we have our single action potential. There's the force we generate. We have our wave summation where the second potential comes in right before the first one is done. So we get that one little bunny hop. Unfused tetanus, multiple action potentials happening in a row, but we can see the bunny hops between one action potential and another. Whereas fused tetanus, we've sustained it, right, completely, and we cannot tell individual twitches. We cannot tell the individual action potentials apart in terms of the force being generated. So, twitch, remember, this is like a single action potential, single electrical shock. We can do this with stimulations, we can do this ourselves. We can apply a slight, charge to something and cause this to happen, but this is that single action potential where that causes a muscle to very quickly contract and relax, right? Because we reached that threshold, right? So we have our stimulus, we have our muscle twitch, right? Here's our stimulus, here's our muscle twitch and response. We increase the voltage, we increase the strength of the twitch to a maximum point. It's not continuous forever, right? Constantly increasing the voltage doesn't mean the muscle twitch will constantly increase either. There's a peak, there's a plateau that we can reach in terms of the strength of the twitch. If we apply separate stimulations, right, a second shock, we do see that we piggyback off the first, right? That's that wave summation that happens. We can see one twitch, we can see two twitches. We see one stimulus here, we can see a second stimulus here, right? And again, that's the summation. The latent period, again, notice here's the stimulus, but we don't reach that peak of the twitch until after that stimulus, because again, there's that latent period. It's not instantaneous. It's very fast, but it's not instantaneous, and so there is a slight gap, a slight delay between the stimulus and the peak of the twitch. And then graded contractions, this is stronger contractions that come from more fibers being recruited until all of the muscle fibers, all the muscle cells are contracting together. And tetanus, we briefly talked about tetanus. Again, we're increasing the frequency of the electrical shocks, right? Which means we decrease the relaxation time between twitches, right? So we see here's the individual twitches. This is 5 shocks per second, right? We can see the peaks and valleys there, so that's why it's called incomplete, because you can clearly determine where one twitch stops and another starts, basically, like where the peaks are. But at some point, there's no relaxation, right? So even here, they're really close together, but you can still tell them apart. Over here, 60 shocks per second, it's a smooth line. That's going to be complete tetanus. There is no relaxation. The contraction is sustained. It's smooth. There's no individual twitches that you can tell apart until we get to the point where fatigue, where the muscle just can no longer sustain itself. It cannot sustain the contraction. I'm going to go through this very quickly because this is, again, review from anatomy, but remember when a muscle contracts, it shortens. We allow movement because when this muscle shortens, it pulls on the bone across a joint. The insertion of a muscle is going to be the attachment point across the joint that is going to move. The origin is on the unmoving part. So like here, we look at the biceps, we see the origin of the biceps is up in the shoulder, right? Like on the scapula and whatnot. And the insertion is down here, like on the radius, right? So the moving part is the forearm, so the insertion is on the moving part, right? It's on we attach onto a bone in the forearm, whereas the origin is on the non-moving part when this biceps contracts in the shoulder. We have different movements depending on how muscles are attached. If it's a flexor muscle, it's going to decrease the angle. So like the biceps is a flexor muscle because it is decreasing the angle, right? So here we have an angle about 90 degrees. If this person contracts their bicep, we can move this arm up, decreasing the angle from 90 to 45 degrees, right? Extensor muscles increase it. So the tricep would be the extensor here because when the tricep contracts, it straightens the arm. So we can go from 90 degrees and straighten out into 180 degrees. So we increase the angle from 90 to 180. We also have agonist or prime movers. This is the main muscle that is responsible for causing a movement. Not every muscle works alone, so we may have synergistic muscles that help the prime mover. And then we also have antagonistic muscles that move opposite. So like the biceps and the triceps, these guys are antagonistic to each other because they perform opposite actions, right? So biceps is going to flex at the elbow, triceps is going to extend at the elbow. We have opposite actions. And so you just pause here if you need to look at this. Again, this is review from anatomy, so if you need to review, by all means do so. But, take some time to remember these terms, extensor, flexor, abductor, adductor, levator, depressor, and rotator.